Have you ever wondered how babies are made? It's a question that has fascinated people for ages. The creation of a new life is a truly amazing process. It all starts with sexual reproduction, where two parents contribute their genetic information to create a unique individual. Imagine a single cell, so small you need a microscope to see it. Now picture that tiny cell dividing into 2, then 4, then 8, and so on. This process of cell division is how a single fertilized egg develops into a complex organism made up of trillions of cells. Pretty cool, right? Here's the really exciting part. Each parent contributes a set of genes, which are like instructions that determine an organism's traits. When these genes combine during sexual reproduction, they create a new combination of traits in the offspring. That's why you might have your mom's eyes and your dad's smile. The journey of a new life begins with fertilization, a truly remarkable and intricate process that marks the very first step in human development. It is here, at the microscopic level, that the story of every person on Earth begins. Fertilization is not just a single event, but a carefully orchestrated sequence of steps that brings together the genetic material from both parents, setting the stage for the creation of a unique individual. Deep within the female reproductive tract, millions of sperm cells begin their journey, but only a few will ever reach the egg. Out of these, just one lucky sperm will successfully penetrate the protective layers surrounding the egg cell. This moment, when a single sperm cell from the father fuses with an egg cell from the mother, is the true beginning of a new life. The fusion of these two cells is called syngamy, and it is a moment of both chance and destiny, as the genetic material from both parents combines to form a brand new set of DNA. The result of this union is a single cell known as a zygote. This tiny cell, barely visible even under a microscope, holds within it the blueprint for an entire human being. The zygote is the very first cell of a new individual, and it contains a complete set of chromosomes, half from the mother and half from the father. These chromosomes carry all the genetic instructions that will determine everything from eye color to height, and even aspects of personality and health. Inside the zygote, the genetic material from both parents merges to form a unique combination that has never existed before, and will never exist again. This single cell contains all the information needed to guide the development of a complex, multicellular organism. The zygote is now ready to begin the incredible process of growth and transformation, as it prepares to divide and multiply. Almost immediately after fertilization, the zygote begins to divide through a process called mitosis. These rapid cell divisions are the first steps in building the trillions of cells that will eventually make up the human body. Over the next several days, the zygote will continue to divide and travel down the fallopian tube toward the uterus, where it will eventually implant and continue its development. From this single cell, all the tissues, organs, and systems of the body will arise, each one following the instructions encoded in the DNA. The process of fertilization is truly the foundation of life, setting in motion the incredible journey from a single cell to a fully formed human being. We know that sperm and egg cells are essential for sexual reproduction. But where do they come from? And why do siblings from the same parents often look so different? To answer these questions, we need to delve into the fascinating world of meiosis. Every second, millions of cells in our bodies are hard at work, dividing and multiplying to keep us alive and healthy. This incredible process is known as cell division, and it's happening all the time, whether you're sleeping, running, or simply sitting still. Cell division is essential for life, allowing us to grow from a single cell into a complex organism made up of trillions of cells. It also helps us heal from injuries, replace old or damaged cells, and maintain the delicate balance that keeps our bodies functioning smoothly. But did you know there are actually two main ways that cells divide? These two processes are called mitosis and meiosis. While both involve a single cell splitting into new cells, they serve very different purposes and have unique outcomes. Understanding the difference between mitosis and meiosis is key to understanding how our bodies grow, repair themselves, and even how we pass on our genetic information to the next generation. Mitosis is the process that allows our bodies to grow taller, heal wounds, and replace cells that wear out over time. For example, when you scrape your knee, mitosis is responsible for creating new skin cells to close the wound. When you grow taller as a child, it's mitosis that's making more bone and muscle cells. Even the constant renewal of your hair, nails, and the lining of your stomach is thanks to mitosis. It's like a biological copy machine, ensuring that each new cell is an exact replica of the original. During mitosis, a single cell divides to produce two new cells that are genetically identical to each other and to the original cell. This means they have the same DNA, carrying the same instructions for how to function. This is crucial for maintaining the consistency and health of our tissues and organs. Without mitosis, our bodies wouldn't be able to grow, heal, or even survive. Meiosis, on the other hand, is a special type of cell division that only happens in reproductive cells, like sperm in males and eggs in females. Unlike mitosis, meiosis creates cells that are not identical to the original cell. Instead, it shuffles the genetic material, resulting in sperm and egg cells with half the usual number of chromosomes. This is what makes each person unique, combining genes from both parents to create a new individual. Without meiosis, sexual reproduction and the incredible diversity of life wouldn't be possible. To truly understand the fascinating process of meiosis, we first need to take a closer look at chromosomes, the essential packages of genetic information that make us who we are. Chromosomes are not just abstract concepts from biology textbooks, they are real physical structures that play a crucial role in every living cell. Imagine them as tiny, tightly coiled threads, each one containing the instructions for building and maintaining an entire human being. These chromosomes are found inside the nucleus, which acts as the control center of the cell. Under a powerful microscope, you can see them as distinct, thread-like structures, especially when a cell is about to divide. Each chromosome is made up of DNA, the molecule that stores our genetic code, and proteins that help keep everything organized and compact. The DNA inside each chromosome is like a long, twisted ladder, known as a double helix. This DNA is organized into genes, which are specific instructions for making proteins, molecules that perform most of the work in our bodies. In total, humans have about 20,000 to 25,000 genes, all packed into these chromosomes. In human body cells, there are 46 chromosomes, arranged neatly into 23 pairs. Each pair consists of one chromosome inherited from your mother and one from your father. This pairing is important because it means we have two copies of each gene, which can provide a backup if one copy is faulty. This arrangement, with two complete sets of chromosomes, is called a diploid set. The word diploid comes from Greek, meaning double or twofold. In scientific terms, we often use the symbol 2n to represent the diploid number of chromosomes. For humans, 2n equals 46. But not all cells in our bodies are diploid. Sperm and egg cells, also known as gametes, are unique. Unlike most other cells, they are designed specifically for reproduction. Their job is to carry genetic information from one generation to the next. Gametes contain only 23 chromosomes, just one chromosome from each pair. This means they have half the usual number of chromosomes found in body cells. This reduction is essential because when a sperm and egg unite during fertilization, their chromosome sets combine to restore the diploid number in the new organism. We call this single set of chromosomes a haploid set, from the Greek word haplous, meaning single. In scientific notation, we use n to represent the haploid number. For humans, n equals 23. This clever system ensures that each new generation has the correct number of chromosomes, maintaining the delicate balance of our genetic blueprint. Remember how we get one set of chromosomes from each parent? The pairs of chromosomes we inherit are called homologous chromosomes. Each chromosome in a homologous pair carries genes for the same traits, but these genes can come in different versions called alleles. For example, one chromosome might carry the allele for brown eyes while its partner carries the allele for blue eyes. Before meiosis can begin, the cell needs to prepare. This preparation phase is called interphase. During interphase, the cell grows larger and duplicates its DNA. It also duplicates a structure called the centrosome, which will be important later on. Think of it like packing a suitcase before a big trip. You need to make sure you have everything you need. Now, the real action begins. The first stage of meiosis is called Prophase I. During Prophase I, the duplicated chromosomes condense and become visible under a microscope. Then something amazing happens. The homologous chromosomes pair up and exchange segments of DNA in a process called crossing over. This genetic shuffling is what makes every sperm and egg cell unique. One special pair of chromosomes determines whether we are male or female. These are the sex chromosomes. Females have two X chromosomes, or XX, while males have one X and one Y chromosome, which is XY. During meiosis, the sex chromosomes also pair up and separate, making sure that each sperm or egg cell receives only one sex chromosome. So, after crossing over, the chromosome pairs line up right in the middle of the cell during Metaphase I. Then in Anaphase I, the homologous chromosomes are pulled apart to opposite ends of the cell. And finally, during Telophase I, the cell divides into two daughter cells, each with a haploid set of chromosomes. The two daughter cells from Meiosis I now undergo a second round of division, called Meiosis II. This time the process is actually pretty similar to mitosis. The chromosomes line up in the middle of the cell, and the sister chromatids, so the two copies of each chromosome, separate. The result is four daughter cells, each with a haploid set of chromosomes. In males, all four daughter cells from meiosis become sperm cells. In females, though, only one daughter cell becomes an egg cell. The other three cells, called polar bodies, are smaller and eventually degenerate. Interestingly, in plants, the polar bodies don't always degenerate. They can sometimes play a role in nourishing the developing embryo. Isn't nature amazing? So there you have it. Meiosis is the incredible process that shuffles our genes and creates sperm and egg cells with unique combinations of genetic information. This genetic diversity is what makes each of us unique. It's also what allows our species to adapt and survive in a changing world. Pretty neat, huh?